All right, check, check, check. Can everyone hear me? Awesome. Thank you. Thank you for all coming today. I appreciate it. My name is Scott Fisher, and I'm the ambassador for HSP in Pennsylvania. We do have some incredible Costco cookies. If you want some to your left, a very healthy lunch sponsored by Drexel. We wanted to do this live because we feel fighting a rare disease is a contact sport. I know a lot of folks were asking, can this be virtual? I think we learned one thing during the pandemic, live is important, right? So thank you all for coming. I know we had folks that driven, I think Ken, you were from Maryland, or you the furthest? Okay. All right, you win a gold star. New Jersey, all right, so thank you so much for all coming. It feels fitting for us to have this in Philadelphia. It's an incredible city, but we have a certain grit in this city. We're the city of underdogs. We've just lost a couple of important playoff games, so we're going to be back next year. And that's a theme that I think the spirit of grit is something that is familiar for all of us that's faced with a rare disease diagnosis. I'm here today as a third generation HSPer, someone who understands that moment when you get a diagnosis and you get in your car and you Google, what did that mean? What did the doctor just say? And I have my incredible wife, Amy, Dr. Google. She looks up everything. And then you get a very humbling diagnosis. There's no treatment. That's awful, right? What do you do? So I hope today is to continue this relationship with all of you in the room to share things as we move forward that help each other. So that's my push for you guys to help come up with ideas for us to stay connected. Other options, family support. For all of us that are here that have HSP, raise your hand. Awesome. Thank you for coming. But there's a lot of folks that didn't raise your hand, and you guys are affected by this as well. So thank you all those that didn't raise your hand, because you're super important. There's power in community, and you guys are all here, and thank you. There's also another word that comes to mind with our session today, and that's one of hope. I looked back on the internet, and there's this thing called Google Alerts. If you have any tech questions, my father-in-law's here for any tech questions. One day, my Google Alerts were highlighted for HSP. I think it was 9 years ago, I communicated with you, Dr. Baas, around some of the initial work that was started. And here we are at Drexel. So I want to just give a round of applause for Dr. Baas and his team. The other thing I'd like to thank him is I just randomly said I'd be an HSP ambassador in December, and I said, hey, would you like to have a session where we talk? And your answer was absolutely. So the fact that we're here today is just a great sign of community, that we have a local expert, local key opinion leader that can help us. Now we're here today. The goal today is to learn, connect, and feel hope for the future and the treatment for our disease. And with that, it's my privilege to turn things over to Dr. Baas, who's going to walk us through the agenda and have his team present the research. And again, thank you all for being here and continue to carry on this sense of hope with HSP. Thank you. Dr. Baas.
Welcome, everyone. Such a pleasure to have you all here. I'm going to start out by giving you a PowerPoint presentation. I think one of the main goals today was to update you on the status of the research that we're doing. So I want to make sure to do that. During my presentation, I will mention the names of people here in our Drexel team, most of whom are here with us. And then I'll close with a little more of an introduction to all their names and faces. And really the rest of the afternoon is all devoted to you to ask questions, anything at all that's on your mind that's not clear, or ideas that you have. I have been emailing with Ken for years and Scott going back. One of the things I'm really committed to is this community. I'm not the type when I get emails from any of you just to let them go by and ignore them. I try to be timely, I try to communicate. I think the same is true of other members of the Drexel team. So we're here for you. This entire event is for you. If anything I say is confusing you, just stop me and I'll explain it better. Okay, so I've been studying hereditary spastic paraplegia for, I think, about 18 years. I'm not a pathologist, I'm not a medical doctor. The way that I began studying hereditary spastic paraplegia is I'm a basic scientist, and I'm interested in a structure inside nerve cells called a microtubule. One of the proteins that regulate microtubules is called spastin. It's a protein that cuts microtubules into shorter pieces so they can move around better. I didn't know it at the time, but the reason why that protein is called spastin is that the gene that encodes it, spast, is the most common gene that's mutated in hereditary spastic paraplegia. That's how I got into studying hereditary spastic paraplegia, because I was one of the very few people in the world studying spastin, so I understood this gene and this protein better than most people. And from there, I really devoted myself to understanding hereditary spastic paraplegia. There are other genes that cause hereditary spastic paraplegia. How many of you here know for sure that you're SPG4? That's spast. How many of you are other genes? A couple people. And maybe some don't know what the gene is. There are over 90 different genes whose mutations cause hereditary spastic paraplegia. SPG4, spast, is the most common. That's the one that we study here. But if you have a different variant, much of what I have to say applies to you equally as well. So understand, the more I talk about SPG4 and spast, many of the therapies, many of the ideas apply across HSPs. I want to start out with just a little bit of genetics. You have one spast gene from your mother and one spast gene from your father. All it takes is a mutation in one or the other, and you got SPG4, hereditary spastic paraplegia. It's an autosomal dominant disease. Now, some of the other HSPs might be recessive, meaning you need a mutated gene from both your mother and father. But SPG4 is autosomal dominant, meaning all it takes is one of them. So if you have one of your parents has a mutated spast gene, that means as a child, you've got about 50% chance that you'll have that mutation. That's the genetics of the disease. What I mean by mutation is a gene has a code, a sequence, nucleotide sequence. ATCG, AATTCG, that's the sequence. All it takes is for one of those nucleotides to be wrong. That's what a mutation is. And a mutation can just pop up randomly for whatever reason. And it happens in all of our genes, that we have mutations in genes. Usually it doesn't matter. In the case of spast, if a mutation arises in that gene, and it could be almost anywhere in that gene, then you have hereditary spastic paraplegia. There are over 200 different mutations. So those of you who raised your hand that you have SPG4 HSP, you may have different mutations. So you have the same gene mutated, but you have different mutations, meaning that incorrect nucleotide might be in some places for some of you and other places for others of you. Whether that matters, whether the disease is more severe or earlier onset, depending on what the mutation is, that's a little bit mysterious. For the most part, it seems about the same, but that issue is ongoing. In your brain, you have neurons called upper motor neurons. So the cell body, the starting point of that neuron is in your brain, what we call the motor cortex. That upper motor neuron puts out what we call an axon or nerve that'll go down your spinal cord, and it'll make a connection called a synapse on the lower motor neuron. And you have lower motor neurons all up and down your spinal cord. And the lower motor neuron is what extends an axon into your hands, your fingertips, your legs. The classic view of hereditary spastic paraplegia is that it's the upper motor neuron that degenerates. And when the upper motor neuron degenerates, it pulls back from that synapse. And now the lower motor neuron is dysregulated. And that's why you have a gait deficiency. Now keep in mind, you have a lot of upper motor neurons and a lot of lower motor neurons. So it may be that not all of your upper motor neurons are degenerating and not all of your lower motor neurons are dysregulated, but some are. And that relates to the severity of your symptoms and the age of onset of your symptoms, which can really vary a lot in patients. I've had people call me who are in their 60s and only beginning to experience the symptoms, whereas other small children are already in a wheelchair. So it can vary a lot for reasons that we don't completely understand. But this is the anatomy. Other tracts, though, can also be affected. There are some individuals who have speech problems. There are some individuals who have cognitive problems, or fecal problems, or urinary bladder problems. So the upper motor neuron is the classic neuron that degenerates in this disease. But it's not only that, and it varies across people. So other tracts can be involved as well, and we're aware of that. In order to understand how a mutation to a gene causes a disease, there are two different disease pathways to keep in mind. One is called loss of function, and the other is called gain of function. Loss of function is now that gene, that SPAS gene, doing what it's supposed to do. It doesn't do it anymore. So now you could be in trouble because you don't have enough spastin doing its work. Remember, you've got half from your father, half from your mother. Only half of it is mutated. The other half is okay. So whether half is enough depends on the gene. Sometimes half is enough, sometimes half is not enough. That's one mechanism, loss of function. It's sometimes called haploinsufficiency. Haploinsufficiency means half is not enough. Is that the cause of hereditary spastic paraplegia? Or is it gain of function? What gain of function is, now you've got that mutated gene, and it's producing a mutated protein. It's not just that that protein doesn't work, it's that it's a troublemaker, it's causing trouble. It can misfold, and it can do things it's not supposed to do. So by gain of function, I don't mean good function, I mean bad function. Gain of toxicity, sometimes it's called. So two different mechanisms that can contribute to this disease. Loss of function and gain of function. If that's not clear, I'd like to show this illustration. Loss of function is the kid didn't show up for class. He's just not there. Gain of function, he's there and he's making trouble. The problem with the troublemaker is it gets worse. And that's what happens in a degenerative disease, it gets worse. Our studies generally show that it's the gain of function spastic protein that's causing the problem. Loss of function may contribute, but we think that it's the gain of function that's the main contributor. That when the spastic protein becomes mutated, it misfolds in ways that result in pathology, toxicity. One of the therapies that everybody always asks about is gene therapy. Gene therapy gets people really excited. And the reason it does is that gene therapy solves both problems. It's called silence and replace. And what you do in gene therapy is you have a vector, which is really just DNA and some other components. And what that vector does is one component will turn off the mutant gene, the endogenous gene inside of you. It will turn off expression from it. And then also as part of that vector is DNA for healthy spastin. So it's silencing the mutant and replacing it with functional spastin. So in theory, it solves both loss of function and gain of function. And we've been working on gene therapy. We just published a paper on it. One of the things to really understand, though, particularly about spastin, is that it's not an easy gene. It's not an easy protein to understand because there are two main forms, isoforms of spastin. One is called M87. That's the microtubule severin protein I told you about. But this gene also encodes a protein, a little bit longer protein called M1. And M1 inserts into membrane. So if you ever heard about endoplasmic reticulum or internal membranes of the cell. So it's really one gene that makes two different proteins. This is a picture of Skanda, who's in the back of the room, and he's a graduate student in Dr. Chiang's lab, and he's very interested in M87 and M1, published a really nice review paper about M1 and M87. So when you think about loss of function and gain of function, it could be loss of M87 function, it could be loss of M1 function. It could be gain of function of mutant M87, it could be gain of function of mutant M1. It could be any of these possibilities. What we think, based on a lot of studies we've done over the years, is that it's primarily gain of function of mutant M1. So it's this M1 isoform that when that isoform is mutated, that's the one that'll misfold and become very toxic and elicit the disease. It's not to say that loss of function may not play a role, it probably does, but what our research has shown is that it's mainly the gain of function of mutant M1. Now, gene therapy is a wonderful concept, as I explained. In theory, it solves everything, but in truth, there are some complications with gene therapy. Gene therapy vectors are driven by a viral vector. It's a virus, we call it AAV9 virus, and that's what conveys the vector into the neurons of your body. And that's kind of complicated. We use a mouse model for the disease, but a mouse has a tiny brain. You've got a big brain. So transducing enough of the neurons in a human brain is a bigger challenge than a mouse brain. And moreover, choosing the right vector that will be harmless to humans is a big deal. If a mouse dies of a vector that doesn't work out, that's a tragedy, but not nearly as much as the human is. So we want to be very careful with viral vectors for humans. There are some other issues. M1, when it's mutated, is not only toxic, it's also very long-lived. So probably those of you who have SPG4, hereditary spastic paraplegia, probably in your spinal cord and in your motor cortex, you've got a lot of this mutant M1 accumulated. And just because that vector turns off the expression more does not mean that that's just gonna go away. It's still there. How long it will be there? A year, 2 years, 10 years, 20 years? We don't know. That's one of the questions that we just don't know. So that's one of the problems with gene therapy, is that the M1 protein may be quite long-lived. Probably even a worse problem, though, is if we express too much spastin, that could be a big problem. With a lot of genetic diseases, it doesn't matter, you can express twice as much, 3 times as much, just so you have enough. Spastin, as I've said a few times, is a microtubule severing protein. Its normal work is to cut microtubules into shorter pieces